Welcome to the note presentation for Chapter 11, Section 2, Probability and Punnett Squares. Remember to fill in your notes for Section 11 too as you listen to this presentation, and you may pause as you need to complete the notes, so don't forget to do that as we go along. So we're going to start with a little learning objective review. Now remember, this is just review. You do not need to write this anywhere in your notes right now. How did Mendel study inheritance in peas? Well, he studied the 7 pea plant traits that he saw, each with 2 contrasting characters. He crossed those plants with contrasting characters and studied their offspring. What did Mendel conclude about inheritance? He concluded that biological inheritance is determined by factors passed from one generation to the next. And what is the principle of dominance? Some alleles are dominant and others are recessive. And lastly, what happens during segregation? And during segregation, alleles separate during gamete formation. So now the learning objectives for this lesson are as follows. There are only 2 of them. Make sure you get these in your notes. Explain how geneticists use the principle of probability, and describe how geneticists use Punnett squares. Okay, so you have all that written down. Let's move on to genetics and probability. The likelihood that a particular event will occur is called probability. How probable is it going to be that something is going to happen? What is the likelihood that an event will occur? We can look at it and we can write it as 4 different ways. So we have these 4 circles here. 3 are red and 1 is white. So the fraction of these circles who are white is 1/4. The percent of these circles that are white is 25%. And the ratio of these circles that is white is 1 to 4. So 3 different ways that our probabilities can be written. So with probability, you can use probability for all sorts of things. You can use it in card games, you can use it in dice games, you can use it to kind of figure out how, what's the possibility that something's going to happen. The way that geneticists use it, okay, how do geneticists use the principle of probability? Your first key concept question. Geneticists use it by using them to predict the outcomes of genetic crosses. So the principles of probability can be used to predict the outcome of genetic crosses. These are the genetic crosses we talked about in the last lesson, where Mendel studied the pea crosses. So when he was crossing different pea plants with different characteristics, and he used that to outline and to start to predict some of these crosses and what would happen. Now, the way that they're looked at is by using something called a Punnett square. And gene combinations that might result from a genetic cross can be determined by drawing a diagram known as a Punnett square. And this is what this diagram looks like. So it's 4 little squares, and the way that it's used, geneticists, how do they use these squares? Punnett squares can be used to predict and compare the genetic variations that will result from a cross. So they're going to take the squares and they're going to start to fill different pieces in. Okay, so the way that we do this is we look at the square here, and we're going to start to put our plants across the top and across the side when we're talking about these pea plants. How we do this is we take the letters that represent this plant. So this T, this tall T, represents the dominant allele for tall. The lowercase letter represents the recessive allele. Now remember, these are going to be the offspring, that first filial generation where we crossed a true breeding tall plant with a true breeding short plant, and we got these hybrid plants that have a tall gene and a short gene. So we take these and we put one across the top and one across the side. And in this example, T stands for tall, capital T stands for tall, and lowercase t stands for short. And we start to cross these together. So it's kind of like multiplication, where we take this T, we separate that in our gamete formation, And we take one, and this goes in both of these squares down here, so the tall t's in each one. We take the smaller t, and we do the same thing. It goes into each of these squares. Then we take it from the side over here, and we take our tall t, and we go across this way, and we take our smaller t, and we go across this way. So these have now been multiplied together to combine to get 4 new plants. The gametes that are produced by each F1 parent are shown along the top and left side. Like I said, we put them along the top and along the left side. The organisms that have 2 identical alleles for a particular trait are said to be homozygous. So if we go back to our original parent plant, they have 2 exact alleles, so we have 2 capital T's for tall or 2 lowercase t's for short. They are said to be homozygous because they have the same traits. Organisms that have 2 different alleles for the same trait are said to be heterozygous. So if we have a capital T and a lowercase t, though we have both the different types of traits, those are heterozygous. The homozygous organisms are true breeding for a particular plant. So if you have homozygous and you cross it with the same homozygous type of plant, you're always going to get that particular trait. And the heterozygous organisms, those are the ones that are hybrid. Those are the ones that are crossed together for a particular trait. All of the plants in this example have the same phenotype or the same physical characteristic. So we have types right here. We have 2 tall plants. They both look tall. And the tall plants do not, however, in this cross have the same genotype or genetic makeup. So even though we have tall, 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 we have the genotype here with a double T, and then a genotype with a tall t, short t, tall t, short t. So these are 3 tall plants, but their genotypes or their genetic makeups are not the same. So if we look just at the tall plants, 1/3 of these tall plants is this tall t, the 2 double t's, the tall t's, and while 2/3 are tall t, short t combined together. And remember, because the tall t is the dominant, that's the one that shows. So now these plants here have different genotypes. So we've got 2 tall plants, one that has the double tall t and one that has the tall t, short t, but they have the same phenotype, and they're tall. So if we had the small t, those would be the short plants. They would have a different phenotype and a different genotype. So the thing to remember is phenotype is what they look like, and genotype is what their genetic makeup is. Now when we talk about probability and that segregation, what we're looking for is how the gametes, how they get segregated, and then how they get combined. So when we look at these 2 tall plants that combine, and they gave us 3 talls with different genetic makeups, they also gave us one of the fourth has this double tall T, 2/4 or 1/2 has 1 allele for tall and 1 allele for short, and then another fourth has the 2 short t's that gives us the short plant. So we'll do more with this in class, but just know that these Punnett squares are the way that geneticists use the probability to figure out what's the probability that a certain trait is going to show up. Okay. Probabilities are also used to predict averages. They can predict the average outcome of a large number of events. However, they cannot predict the precise outcome of an individual event. So even though you can say, according to the Punnett square and according to my predictions from this, that 3/4 of the time you should get a tall plant, it is still very possible that you could get more than one small plant or less of the tall plants. But what happens as the larger number of offspring, the closer the resulting numbers will get to expected values. And this happens when you roll dice as well. If you expect, or if you flip a coin, for example. So if you flip a coin and you got a 50% has heads, 50% has tails, the more you flip that coin to get that outcome, the more it's going to come when it's closer to the 50%. If you flip a coin twice, you're probably not going to get one heads and one tail, but the more you flip it, the closer you're going to get to that 50%. Okay, let's finish up this lesson with the learning objective review for 11.2. And the first question, remember, these are the ones you need to write on your own. They will not be shown on the screen. So you need to listen to what I'm about to tell you or go back in your notes and find the answers. How do geneticists use the principles of probability? They use probability to predict the outcome of genetic crosses. And how do geneticists use Punnett squares? They use them to predict and compare the genetic variations that will result from a cross. Okay, well, this has been the presentation for Chapter 11, Section 2. Make sure that you have all of your notes filled in for 11.2 before we put them in our notebook. And if you need to rewind and pause to get everything filled in, please do so. And we will see you next time.